Hello, my name is Rakan Naboulsi, and I will present to you the association of hair whorls and behavior in the standard-bred horses. As an outline for my presentation today, I will present the scientific content by starting with some background, and then I will move to the materials and methods to describe what we have done to achieve the results and our final conclusions. Then I will, because today is not only about the science, but about the technical aspects, so I will talk about some quality controls, the limitations of the kit that we used, and the filtration parameters, and then I will mention the strengths of the kit. So background, we know that animals have their own personalities and that these personalities, they affect their athletic performance. And we also know that behavior is heritable. Hair whorls, they form during fetal development and they emerge from the ectoderm. And interestingly, they are used by horse trainers to select the horses for what sport they will be training them. Genes also were found to affect the position of the hair whorls, and the same genes were found to be associated with psychiatric traits in humans, such as depression and schizophrenia. And the hair whorl is also heritable. For example, the heritability was found to be 0.8 in Polish conic horses. Hair whorls, they have different shapes, and I here will present 4 of the shapes. One is clockwise, the other is counter, radial, and there is a type that looks like a feather. They can be situated all over the body, but today I will talk about those that are placed on the face. And then you have the right side of the face, the left side, and the medial. The horse to the left here, it has one on high up on the face, and this is the middle, and this is low, and this is medial, right and left. So this is 1 and this is 2. Whereas this one here, this horse here, it has a single feather that's located to the right side of the face. Our aim is to investigate the association between hair whorls and different behaviors. So, for example, if the horses are to pass in a path, if they have this shape of the hair whorl, will they choose this path or will they choose the other path? This is, for example, one of our aims. Materials and methods. For the survey, what we have done is that we have asked questions for the horse owners about 15 different traits and a lot of questions per trait. And we asked about things such as the nervosity, the concentration, the cooperation of the horse, and we also asked about the hair whorls, and we had a person in the lab that went out to visit the horses and investigate how does their hair whorls look like. We have a specific protocol to be followed to describe the hair whorls, their type and their shape, and we identified about 19 different types of the hair whorls. Then we have done an association, a statistical test to study the association between the behavior and the hair whorl type. And for that reason, we used about 190, where some of them were removed, and we kept 175 standardbred horses that are shown in the results that I will present today. But currently, I am analyzing more horses, so we will end up with having about 300 horses in total in our research. We extracted the DNA from the tail hairs. We plucked the hairs from the tail, including the root, and we used the Chelix method to extract the DNA. We measured the concentration using qubit because it has better accuracy than some other methods. The whole genome sequencing library prep was done according to the manufacturer's protocol. Sequencing, we have performed that on Illumina Novasec 6000 instrument, and we used an S4 flow cell, which gives about 2x coverage per horse if we use a full plate on one cell. Then I have analyzed the data when they came to me, and I aligned them to the EQCAP3. Until I got the BAM, I used the GATK best practices pipeline, which takes me from the FastQ to the BAM files, which are the analysis ready BAM files. Then after that, I converted the BAM files to a VCF file, and there I calculated the FST. But then I realized that there are things that I didn't like in the analysis, so I used another software that is better in analyzing low pass sequencing. So what I did is that I used the final BAM files, the analysis ready BAM files from the GATK, but then instead of going through and making the VCF file, then I use those BAM files as an input for this software, which is called PCA and GSD. And then I calculated the allele frequency likelihood, and for my two opposing phenotypic groups, I calculated the delta allele frequency. And delta allele frequency is usually highly correlated with FST. As a result, we saw that the localization of the hair whorls on the face, if it's to the right or to the left, is statistically significantly associated with nervousness. The number of facial hair whorls is associated with their ability to learn how to compete, and the orientation of the hair whorl, if it's inclined or vertical or diagonal, then it is associated with their ability to focus and their appetite after competition. I show this as an example of the results. We have more of these Manhattan plots, but I will only show this one as an example. And here I show the delta allele frequency, where we compared horses that have one hair whorl versus two hair whorls, and we did this analysis in windows of 50 kb size. The position of the chromosomes here is changed, except for the chromosome unknown that you can see it's here to the right, but then the rest of the chromosomes, they are shuffled, so this is not chromosome 1. And here we find a significant window. This is the significance threshold, Bonferroni corrected, and we think that this is statistically significant between the horses that have one hair whorl or two. So as a conclusion, we conclude that hair whorls can be used to identify horses with specific personalities. We can choose horses for different tasks based on their personality and also to predict their behavior. So now I move on to the technical part. Using this kit, the Twist 96 Plex, we extract the DNA from horses, we pool them at the very first step together in equimolar amounts, and then we prepare the library and send it for sequencing. What we get from the sequencing facility back is FastQ files, and I check the quality of those files by using the FastQC software. And as you can see, those two are the read 1 and read 2 of the same sample, and they are representative of the other samples. That's why I chose them. So the quality of the data looks good, but the drawback, or one of the limitations of the kit, is that although we try our best to measure the concentration of the DNA samples accurately and pool them in equimolar amounts, but we still have some variation in the amount of reads that we get per sample. So some samples we got about 0, and other samples we got around 12 million reads, and those are for one plate that we used as a first step as a pilot test. So yeah, this is one of the limitations that I think are of the kit. Then coming back to the default pipeline that people like to use, which is the GATK best practices. I went, as I said in the beginning of the lecture, from the BAM files, the analysis BAM files, to the VCF, and in the VCF we have a lot of different parameters. And because we are working with a non-model organism, then we cannot follow what is done for humans and mice, which is the VQSR. So we need to do something that is called hard filtering to remove variants with low quality. Here, this is the method shown by GATK and their recommendations. So when they use a sample, a human sample, and they look at it, this is how it looks. Those here in the red area are the bad reads that need to be filtered out, and those are the blue, the good reads that need to be kept. So for this, they use a filter of QD less than 2, so everything less than 2, it will be removed. And this is their non-filtered sample, or yeah. My samples look like this, so it is a little bit different. They have two peaks that you can see here. This peak refers to the heterozygotes, and this one corresponds to the homozygotes in their samples. I have them opposite. I have double amount of the homozygotes as compared to the heterozygotes. So that made me think, why is that? This is not really accurate. So I wanted to look on other parameters. Then I looked at the Fisher strand, and the same. The red here is the bad reads that need to be removed, and this is the good reads that need to be kept. And here they have a filter of FST greater than 60, which is something here. They will remove it. So they will remove, they will remove a large part of the bad reads, but they will not affect their good reads. When I wanted to do the same thing, well, my x-axis doesn't even reach 60. So this is something else that made me think there is something that I need to change. Here I have nothing. It looks good. And here, well, what they have is MQ less than 40. It needs to be filtered out. But if I filter my samples at 40, then I will remove about 30% of my samples, which is something that I don't want if they are not correct. So what I realized is that GATK works perfectly for reads that have a high confidence in the genotypes, for example, but with low quality the confidence does not exist. For example, if you look at this read here, it is a single read, and this SNP that should be here is supported by a single read. So if this is a nucleotide A, GATK will consider it to be homozygote A. But that's not accurate, because what if it is a heterozygous and we couldn't see the other read just because we have only one read? And even if we have two reads like this one here, those two reads, even if they belong to the same nucleotide, GATK will consider them to be homozygote, but they are not, maybe. We are not sure. So the confidence is low because we have two reads, and the confidence will start to be good at 10x coverage, not more. So GATK has certain problems if it's followed with low pass sequencing. So for this reason, I used the PCAN-GSD software. That is better. So that software doesn't go with the confidence of the genotypes, but it calculates the likelihood, so probabilities, that this horse has more probability of being homozygous reference, suppose, than homozygous alternative, because it has one read that is a reference read. So finding that software solved this problem, and then we could use it to analyze this data. Now I move to the strengths of the kit. What I like about this kit is that I can sequence the individuals each one alone, not in pools, and the price will be low, but then afterwards I can reshuffle my samples and create different groups that I can analyze later. This, for example, here I show that I'm comparing the horses that have a red hands, red front legs, with blue ones. But then later on in the future, I might think of something else, so I would like to maybe change my groupings, so I can just change them, and now I have the green versus black hind legs. So this is something that I think it's nice with this kit, and one of the reasons that I used it. This is the end of my presentation, and I would like to thank my PI, Gabriela Lindgren, and my colleagues Maria Wilbe and Victoria Saxe. Thank you so much.